So this is our training conference. We're going to start out with the basic science and clinically oriented basic science with Dr. Dustin Sulak. He's going to teach endocannabinoid physiology and cannabis pharmacology. And as you know, I hope all of you know, the endocannabinoid system is the most important part of our body's physiology as it relates to how we use cannabis, and it's essential to replace that paradigm that many of us have been taught, that cannabis is a drug of abuse, and replace it with this really core knowledge that it is a regulator of the endocannabinoid system. So that's first. Then we'll be followed by Cynthia Norcutt, who's an attorney and a nurse, and she specializes in cannabis medical legal practice here in Missouri, correct? And we thought that would be also essential for you to understand the liabilities and or ways to navigate the system if you're interested in doing that in this state. And then we're going to have a short break for about 10 minutes, hopefully, only. And then I'll be coming in with a review of the top 10 important conditions to know about clinically and some specifics about Missouri. And then we'll have a short 15-minute Q&A. We'll be done at 5. So thanks for being, oh, one more thing. If you have registered to be here, which I hope most of you have, you'll be eligible to get CMEs, or CEUs, sorry. If you have not registered, you will not be eligible to get CEUs. If you would like CEUs and you have not registered, please go up to the front desk and you can actually pay your dues after this wonderful presentation. Thank you. And here's Dr. Sulak.
Welcome, everyone. Glad to be here with you all in person. For a lot of us, it's been a while. So we're going to dive into endocannabinoid physiology and cannabinoid pharmacology. And the format of this is intended for clinicians. But I know one of the great things about Patients at a Time is we always have a bit of a mixed audience. I think that's one of our strengths. And so I'll also do my best to translate this information to make it accessible to everyone, like the plant is accessible to everyone, and to help us all share, and like Deborah said, to have a solid foundation for the rest of the conference over these next couple days. So a quick potential conflict of interest disclosure. I am an equity owner and employee of a company called Healer. We provide free patient education at healer.com. We also have cannabis and hemp products. We have paid industry training. We also do some consulting and extraction and formulation technology. I am formerly, within the last year, a paid scientific advisor for an analytic company called Forian. Also unpaid member of the board of directors of Society of Cannabis Clinicians. And if you're a clinician and you're not familiar with the SCC, I strongly encourage you to check out our group, which is getting more and more active. We have a lot of great offerings. And then the author of a book that came out last year, Handbook of Cannabis for Clinicians. This is what we'll be covering in the next hour or so. We'll do an overview of the endocannabinoid system, starting with the receptors, then getting into the compounds that our bodies make to target those receptors, and we'll tour a few of the systems in the body, the nervous system, including pain signaling, the immune system, and some of the effects in GI and metabolism. Any of these bullet points could easily be a whole day of discourse if we chose to, but this will be a good overview. We'll talk about what happens when the endocannabinoid system may not be functioning optimally, and then we'll look at some of the compounds from the plant and how they interact with our endocannabinoid system and other targets in the body. So we'll start here with a partial list of health conditions for which there's some clinical evidence that cannabis or the compounds in cannabis can be effective. And if you look at this list, you'll see examples of disease states from basically every field in medicine: psychiatry, rheumatology, gastroenterology, neurology, and so forth. And I think that when we, any clinician that's kind of gone through conventional training and sees a list like this, we think, oh, this is, BS. How could this possibly be true that one plant could do so many different things? It seems very unlikely. And that was an important question that was asked for a long time. The answer came in the late 1980s and early 1990s when researchers investigating the action of THC in the body discovered something that later became known as the endocannabinoid system. So let's talk about the endocannabinoid system. It's a homeostatic regulatory system that's present throughout the body. Its main job is to help maintain resilience and balance in the state of stress and changing external environments at the cellular level. Now, there's been an enormous amount of research into this system. If you look at PubMed citations back in 1993, there were 10 citations for the term endocannabinoid. This year, over 11,000, almost 12,000. Just these are scientific papers that mention the term. And so if you think about that, that's like one paper every other day for the last 30 years, just about. It's a lot of information, a lot of investment in resources of time and energy and money into investigating this system. And why? Because it's such an excellent target for disease. Because it's this global homeostatic regulator of the other systems in the body. If we learn how to interface with this system, we can use it to affect all of those different disease states, including refractory conditions from every field in medicine. And that's what we do. So let's take a little deeper look at the components of this system, beginning with the cannabinoid receptors. These are the CB1 and CB2 receptors. And as you can see, if you're familiar with cellular receptors, these would be located in the outside of the cell membrane, and they're proteins. These have 7 passes across the cell membrane, characteristic of the G protein coupled receptor family of receptors. Here you can see a 2-dimensional image and a 3-dimensional image. The CB1 and CB2 receptors are very similar. CB2 is in gray in the 2-dimensional image and CB1 in black, but you can see there's some differences, and those differences show up in our tissues. So CB1 is primarily located in the central and peripheral nervous systems, but actually all over the body as well. I mean, both of these are widely distributed. You can see that CB1 is present in the connective tissues like the fascia, adipose tissue, skeletal muscle, smooth muscle, liver, lungs, and so forth. CB2 is primarily located in immune cells, also found in the bones, also found in the CNS. It's a bit controversial whether CB2 exists in the healthy central nervous system, but certainly when the nervous system becomes inflamed or injured, we see upregulation and production of CB2 receptors there. And then also in the enteric nervous system, the nervous system of the gut. In addition to these outer cellular membranes that I just described, the CB1 and CB2 receptors have been found in other places inside the cell, including on the mitochondrial membrane, where they seem to have a role in regulating ATP or energy production and cellular respiration. They're found in other organelles, the endoplasmic reticulum, endosomes, lysosomes, and cell nuclei, and we don't really understand what they're doing there, but probably something. Now, if you take a look at PET CT combination of the human brain, or PET MRI, excuse me, you can see there's a distribution of CB1 receptors really all over the place there. There's some areas that have more CB1 receptors. So, for example, would be like where the red areas are, so the limbic part of the brain, some of the motor centers of the brain, cognitive areas of the brain. All of these areas where we see the most CB1 receptors correlate with functions that we see altered when people consume THC, like motor function or memory function or mood and emotion. Here, and what I wanted to mention here is that if you take all of the other different G protein receptors or cell membrane receptors in the brain, including, say, like dopamine receptors, serotonin, GABA, glutamate, all those different neurotransmitters, there's more CB1 receptors in the brain than any of them. It's truly a high population of this protein in the brain. CB2 receptors found throughout the periphery. Here you can see them in high abundance in the liver, in the lymphatics, skeleton, the spleen. And so, again, like this is just a system that is ubiquitous throughout the body. It's really found everywhere. And when we interface with the system, we're interfacing with our whole system, our whole body, maybe more than our body, mind, spirit. And it's a very old physiologic system. So the CB receptors have been around in animals for around 600 million years. If you look at a very primitive chordate, the sea squirt, it has a CB receptor that's almost identical to the human CB1 receptor, which means this has been important. This is something that animals have been doing for 600 million years, and it has not been selected against, it's been selected for. So this is a high level of evolutionary conservation. And compared to 600 million years, the cannabis plant is believed to have evolved around 34 million years ago. So animals were using cannabinoids long before cannabis came around. Now, we don't just have the CB1 and CB2 receptors so that we can respond to cannabis. There's probably some endogenous reason that we have these receptors, and that was discovered in 1992 and 1995, the endocannabinoids, as they're now called. The first one, anandamide, Sanskrit word ananda, that's what it was named after, and later 2-arachidinyl glycerol, or 2-AG. So these are pictured in their molecular forms. They look a bit different than THC, for example, but from a molecular structure, but they have similar function. In the nervous system, they serve as retrograde messengers, and I'll show you more in subsequent slides what that's all about. Elsewhere in the body, they're produced as autocrine and paracrine mediators, which means a cell will produce these. They'll act either locally on the receptors on that same cell or on neighboring cells, and then they'll be degraded. Some of them do circulate in the body, and it's believed that there are some more global systemic endocrine effects of the circulating endocannabinoids. As I mentioned, they're synthesized on demand. Their precursors are omega-6 fatty acids, which are essential fatty acids. We need to get them from our diet. And they're rapidly degraded by these two enzymes primarily, Pha and MAG lipase. And so interacting with these enzymes could change the levels of endocannabinoids in the body and may have some therapeutic effects. Now, this is just a summary lecture. I want to point out those are the two main endocannabinoids. There are many other endocannabinoid-like compounds whose function is not very well understood, but growing all the time, and it's just not worth us talking about this. What I want to leave you with is the trends of how the system functions in the body, knowing that you have not seen all the details, all the specifics. The endocannabinoids don't just target the CB1 and CB2 receptors. There's many other targets in the body where these compounds act. GPR55, which is prevalent in the brain. The TRPV1 receptor, this is the so-called capsaicin receptor after it's the compound in hot chili peppers, which stimulates it, useful for treating pain and inflammation. The PPARs, these are receptors that are located on the cell nucleus, so not on the outer membrane, but inside the cell. These are receptors that control the turning on and turning off of genes. And primarily, it's the ones that cannabis affects have to do with pain and inflammation and metabolism. The cannabinoids also target voltage-gated ion channels and ligand-gated ion channels. So you can see there's a very complex system. Now let's go to the nervous system now. We're in the brain. This is a very simplified diagram of what's happening in the synapse, the communication zone between two neurons. And you can see in red what you're probably familiar with, this idea of an anterograde transmission, where basically the presynaptic nerve is depolarized. These neurotransmitters that are stored in vesicles, they're released. Those act upon the postsynaptic cell, and that causes an effect in that cell. And that's what we're kind of used to seeing. The endocannabinoid system works the other way. The endocannabinoids are not stored in vesicles. They're synthesized on demand. Instead of going forwards across the synapse, they go backwards. And when they get there, that's where they find the CB1 receptors. So why are cells doing this? What's the function of this retrograde signaling? Well, we can start here with a couple pictures. This would be an example of an excitatory synapse. What you're seeing here is that after that action potential came down that presynaptic nerve, calcium came into that nerve terminal that caused the release of glutamate, an excitatory neurotransmitter. Eventually, that's going to stimulate the glutamate receptor on that postsynaptic cell. That's going to open some calcium channels, and that influx of calcium is going to create a downstream effect in the postsynaptic cell. Eventually, when that signaling becomes strong enough, the postsynaptic cell is going to synthesize an endocannabinoid, in this case 2-AG, and that's what's going backwards across the synapse to stimulate that CB1 receptor, which closes the presynaptic calcium channels and prevents further release of that neurotransmitter, glutamate. So this is called depolarization-induced suppression of excitation, meaning that the cannabinoid system is kind of putting the brakes on, turning off that excitation that's coming from that presynaptic nerve. Now, if we change the diagram from glutamate to GABA, this would be called depolarization-induced suppression of inhibition, because GABA is an inhibitory neuron. So you're starting to get the picture that this is a signal from the postsynaptic cell to the presynaptic cell saying, wait, don't send any more of that over. Now, if we have stimulatory and inhibitory neurotransmitters and the same system controlling them, you begin to see how this is really a balancing act in the body, that this is functioning all the time to create balance in our nervous systems. Not only for the purpose of regulating neurotransmitter release, the endocannabinoid system is, I should say not only for that purpose, but as a part of that purpose, the endocannabinoid system modulates neuroplasticity via multiple mechanisms. On this slide, you can see the two I mentioned earlier, depolarization-induced suppression of excitation and inhibition. The endocannabinoid system also modulates neurogenesis, the actual growth of new nerves, and two other processes involved in neuroplasticity, long-term potentiation and long-term depression. Now, what's neuroplasticity? We have it in our lives all the time. When you watch a baby learn how to move its body, or when you watch somebody recover from a stroke who couldn't talk or walk and they learn to do that again. I mean, just millions or maybe billions, I don't know how many new neuronal connections are being produced rapidly during those times. More subtle types of neuroplasticity, like subconscious acquisition of some behavioral traits from the people you spend time with, or emotional traits from the people that you're close with, gaining a new skill. All of these are neuroplastic events. The endocannabinoid system is also highly involved in neural protection, meaning protecting the brain from injury. Anandamide and 2-AG are endogenous neuroprotective agents that we produce upon chemical and mechanical trauma. So in basically every type of brain injury that's been studied, it's been shown that mammals at least, and it's believed that humans are the same way, will produce very high levels of these endocannabinoids because they protect against that injury, prevent the extent of the damage, and can speed the healing. Delta-9-THC, CBD, anandamide, and 2-AG, as well as the synthetic cannabinoid HU210, have all been shown to decrease glutamate excitotoxicity. One of the things that can happen in a brain injury is that more and more glutamate can be released from the injured and dying nerve cells. That becomes toxic to the surrounding cells, and then they start to leak glutamate, and things just get too excited in the brain. All of these compounds have been shown to reduce that, to reduce seizure activity, to limit the size of a stroke after it happens. Cannabinoids have also been shown to be effective at reducing and preventing perinatal brain injury. So from when babies are born and they have a lack of oxygen, it's a really opportune time to protect the brain and really change the course of somebody's life. Moving on, in the nervous system still, we'll talk about the autonomic nervous system. Autonomics are the part of our nervous system function that we don't usually have to think about. We have two branches of the autonomic system, the sympathetic and the parasympathetic. Sympathetic is thought of as the fight or flight nervous system, parasympathetic, the rest and digest. That's a major oversimplification. But anyways, in the sympathetic nervous system, the CB1 receptors, when they're stimulated, they inhibit norepinephrine release. They've been shown to dampen sympathetically mediated pain and to modulate the HPA and the HLN axes, which are the way that the brain communicates with the adrenal glands to produce things like cortisol and indirectly adrenaline. In the parasympathetic nervous system, CB1 again is reducing elevated activity, which is thought to be the mechanism of action of the anti-nausea effects of cannabis, or at least of the THC in cannabis. And so here you can see again like two examples of when the body's doing something excessive, too much fight or flight activity, too much rest and digest activity to the point of salivating and almost about to vomit. The endocannabinoid system will suppress that excessive activity. Moving on in the autonomics, a major part of what the autonomics do is control our blood vessels and our cardiac function. There are CB1 receptors in the muscle of the heart, which can have differing effects on heart rate and cardiac contractility. It can increase it or decrease it. Vascular tissue has CB1 receptors. Activating that usually causes vasodilation. Those are both via the relaxation of the smooth muscle cells, as well as a nitric oxide mechanism from the endothelium. The cannabinoids have been shown to produce antihypertensive or blood pressure lowering effects in humans. And there's been animal models of heart attack where administering cannabinoids like THC in tiny doses can prevent the extent of the damage. Now let's focus a little bit on the endocannabinoid system and pain. We'll start with preclinical models, animal models, studying, activating the system, often with synthetic cannabinoids, but sometimes with endocannabinoids or plant-based cannabinoids, can have activity in all the different types of pain that are studied. So the ECS isn't particular to acute pain or chronic pain, inflammatory pain, or neuropathic pain. It seems to work everywhere. And early on, when researchers were looking to discover synthetic compounds that could stimulate CB1, they used this tetrad test to evaluate these different compounds, basically in animal models, mouse models. And one of the criteria for a drug that might be a potential CB1 agonist was analgesia. So it's this very basic aspect of what CB1 stimulation, and CB2 for that matter, can produce. The anti-nociceptive or anti-pain signaling effects of cannabinoids involve many mechanisms. We'll just cover a few in these slides, or actually let me remind myself if we cover them. I'll cover a few on this slide. So the descending pain inhibitory pathway, this is a way for our brain to send a signal down to the rest of the body to say, stop sending so much pain, turn that pain off. And that's really important when we're, say, in a situation where we have an injury and the pain is overwhelming, but we have to remove ourselves from that scene, from that situation to get ourselves safe. So we have to be able to turn that system on, but it also has to not be on all the time, or else it could be dangerous for us. By stimulating CB1 receptors in that system, it kind of takes the brakes off of that system and lets it do a little bit more to inhibit pain. There's also cannabinoid receptors peripherally in the terminal of the pain signaling nerves. So like say you stub your toe and it's hurting, there's cannabinoid receptors down there. And one of the mechanisms that prevents that acute pain from turning into long-term pain or chronic pain is the function of the endocannabinoid system to decrease that pain signal a little bit, suppress some of the activators and sensitizers of that neuron. The cannabinoid receptors are in the dorsal horn of the spinal cord where the pain is heading up to the brain. Stimulating them there also suppresses that signal. And then supratentorial sites referring to aspects of our brain that not necessarily feel the pain when it comes to the brain, but interpret the pain. So our kind of emotional response to the pain, the bothersomeness of the pain, and the behavioral aspects of our experience of the pain all can be mediated by the cannabinoids. It's also been shown that cannabinoids and opioids can work really well together. Our body actually uses them together. We produce endogenous cannabinoids. We also produce endogenous opioids, the endorphins and the enkephalins. And it's been shown that these receptors for the opioids and for the cannabinoids are present in the same areas of the brain that control pain signaling, and that they interact with each other. It's been shown in numerous animal studies that administering cannabinoids and opioids together results in a greater than additive anti-pain effect. meaning it's greater than the sum of its parts. There's a synergistic effect between cannabinoids and opioids. Our bodies know this. We're using this in our strategy for pain. It might make sense if we're thinking about that strategy from an external medicine point of view. And I know that opioids have gotten a really bad rap because of basically what humans have done to them, even though it's a plant that was put here on Earth like cannabis and or evolved here on Earth like cannabis, and has a lot to offer our species in terms of reduction of suffering and compassionate use of that medication. So it makes a lot of sense to use cannabinoids and opioids together. Here's just some review data from 19 preclinical studies on the co-administration of these compounds. What they found was that the effective dose of morphine was 3.6 times lower when administered with THC compared to when it wasn't. The effective dose of codeine was 9.5 times lower when administered with THC. If we could see even a fraction of this synergy in our patients when we treat them with opioids, that would be really exciting. Well, the answer is it's there. Like study after study, most of them are observational and not the so-called gold standard randomized control trial. But again and again, we see this data coming out showing that patients with chronic pain that are using opioids that initiate using cannabis, somewhere around 40% of them are able to stop their opioids. Another 40% are able to reduce their opioid dosage and improve their symptoms and improve their function and quality of life. There's just so much data on that already. So in summary, what did we learn about endocannabinoid system in the nervous system? Retrograde synaptic transmission, remember that backwards across the synapse, a major role in neuroprotection, a role in neuroplasticity and learning, the regulation of the autonomic nervous system. And if any of you think of any of your patients and any of the conditions that any of your patients have, is there an influence of the autonomic nervous system on that condition or the symptoms it produces? Of course there is. Anti-nociception or anti-pain signaling, and synergy with the opioid system. Now let's move on from the nervous system to embryology. CB1 receptors are detected in mouse embryo as early as the second day of gestation. It's also been shown that for the blastocyst to implant into the endometrium, there has to be appropriate levels of anandamide there, not too little or not too much. So this endocannabinoid system is highly involved in the very beginning of life. The proliferation and differentiation of stem cells are shaped by extracellular cues provided by endocannabinoids. So this system has to be functioning for our nervous system and for the rest of the embryo to develop appropriately. This is a falsely colored image of a 6-day-old human embryo implanting itself into the wall of a womb. It's just incredible to think that that's what we all were at some point, and that initial function of finding and connecting with the womb was at least in part mediated by the presence of endocannabinoids in suitable levels. Was there a question on that point?
Well, I heard at the other end that the very first thing that the baby has, because it doesn't have any cellular memory, is the CB1s and the CB2s activated so that it will suckle the breast milk.
Right, so a role in the suckling. So, and again, we see this kind of metaphor in endocannabinoid function. You'll see it again when we get to the metabolism part, but there's this kind of nutritive aspect to it, right? So blastocyst implanting on the endometrium and allowing that nutrition to flow. And then if you block CB1 receptors in newborn mice, they fail to suckle, they'll die. It's a part of our hunger response, part of our nutritive response and our connection to life. Endocannabinoids are very active in the bone, regulating osteoblasts and osteoclasts. So these cells are the ones that build bone and break down bone, and this remodeling process of bone is required throughout one's life to have healthy and strong bones and to heal from injuries. CB2 stimulation in the osteoblasts and osteoclasts seems to have opposite effects. So it'll suppress osteoclast activity and stimulate osteoblast activity. CB1 receptors are also present in the sympathetic terminals that innervate the bone, and it's the norepinephrine that's released that can suppress bone growth. This is thought to be one of the mechanisms in the pathophysiology of scoliosis. If there's this kind of asymmetrical sympathetic activity, the bones can grow asymmetrically. Oh, where are we going? Immune system. So T and B lymphocytes have CB2 receptors. There tends to be stimulation or modulation, we should call it, because it can increase or decrease the activity depending on what cell type and what the situation is, but controls or modulates the proliferation, so the growth of these cells and the death of these cells, modulates macrophage-mediated killing of sensitized cells, inflammatory cytokine production, and again, this would be suppression of excessive activity. Immune cell activation by inflammatory stimuli modulates chemotaxis, or the movement of immune cells based on chemical gradients, and the migration of inflammatory cells in the body. Now, there's trends in the immunomodulation with the stimulation of the CB2 receptors. There tends to be a decrease in the Th1 type of cytokines, decrease in metalloproteinases, and increase in the Th2 types of cytokines. And these are often associated with the problematic effects in autoimmune and rheumatologic diseases, and then also shown to increase subsets of Bt and natural killer cells. Moving on to the digestive system, CB1 receptors modulate activity in the enteric nervous system in a similar way to the central nervous system. They can have an influence on gastric acid secretion, the tone of the lower esophageal sphincter between the esophagus and the stomach, intestinal motility, how fast things are moving, how sensitive the gut is to pain, and how inflamed the gut is. Now, CB2 receptors are also located in the gut. They are where you would expect them to be. They're in the lamina propria, in the plasma cells, and in the activated macrophages. This is the way that one way that the gut modulates its immune system. And they're so involved in the inhibition of inflammation, visceral pain, intestinal motility, especially more active in the inflamed gut. Endocannabinoid system controls hunger and metabolism. As I mentioned earlier, breast milk contains endocannabinoids, and these receptors are important for suckling in newborns. The endocannabinoid system modulates the metabolism of the cells via all of these other hormones, which have gotten a lot of attention in the last decade or two for regulating energy production and energy liberation, appetite, and satiety. Now, obesity can lead to an overproduction of the endocannabinoids by adipocytes, by the fat cells. And this can actually cause a problem that drives CB1 receptors into a feed-forward dysfunction. So it's thought that in obesity, with an overproduction of endocannabinoids, you can actually start to build tolerance, as almost analogous to building tolerance to THC. And this would downregulate CB1 receptors and cause a dysfunction in the endocannabinoid system that would then be less able to correct some of the problems there with obesity. So you can potentially get into a feed-forward dysfunction that contributes to metabolic syndrome, whereas that same system could be a target to treat metabolic syndrome. Now, it's interesting because THC serving as a partial agonist at the CB1 receptor, we'll talk a little bit more about that later, it could theoretically correct both of those situations. Someone with obesity that has a downregulated endocannabinoid system, or someone in another situation that actually could use some stimulation of that system. In summary, the endocannabinoid system is widely distributed throughout the body. The primary function of the endocannabinoid system is cellular homeostasis. Our understanding of it is incomplete, emerging, and suggests significant complexity. We will be diving into that complexity over the next couple days, and I'm looking forward to that. Manipulation of the endocannabinoid system may provide effective treatment in a wide variety of diseases. And to drive that point home, I'm going to read this quote from a 2013 paper from two prominent authors at the NIH. Modulating endocannabinoid system activity may have therapeutic potential in almost all diseases affecting humans, including obesity, metabolic syndrome, diabetes and diabetic complications, pain, neurodegenerative, inflammatory, cardiovascular, liver, gastrointestinal and skin diseases, psychiatric disorders, cachexia, cancer, and chemotherapy-induced nausea and vomiting, amongst many others. Just incredible. So having learned what you know about this system, it's an important question to ask, what happens when it may not be functioning right? Like, is it really doing its job to the best of its ability in all of us? What does that look like? Well, to begin with, there's been a surprising amount of research on the polymorphisms of the genes that code for the CB1 receptor especially. So there's different versions of this gene. Some of us may have little mutations that, based on outcome research, might make us more susceptible to these variety of conditions. Schizophrenia, alcohol dependence, changes in body mass index, obesity, ADHD and PTSD, happiness, serum lipid profiles, and so forth. So looking at large populations of individuals that have these genetic polymorphisms in the CB1 receptor, one can potentially predict outcomes. And in those people that have these conditions, finding a way to restore balance to the endocannabinoid system might be a therapeutic strategy. Ethan Russo has published 2 papers on this topic, the most recent in 2016, Clinical Endocannabinoid Deficiency Reconsidered, and he visits migraine, fibromyalgia, irritable bowel syndrome, and other treatment-resistant syndromes in this paper. Some of the thing that ties them all together is this hypersensitivity. These are all conditions where conventional medicine does a very poor job of treating them, doesn't really understand their cause. They're kind of too complex for the reductionistic black and white model that's prevalent in kind of the pharmacological approach to medicine. And so they're treatment resistant. But hypersensitivity is often a feature of people with these conditions, and cannabis seems to really help bring that into balance. Similar story with autism spectrum disorder, in which there can be also a lot of hypersensitivity. This is one example of a couple of papers that have shown that children with autism are likely to have lower levels of the endocannabinoids than their neurotypical controls or matched children that are neurotypical. Okay, so that's the endocannabinoid system, a great target for pretty much any disease, something that might not be functioning well in all of us. Let's take a look at one way of interacting with it. And of course, my favorite way, and I think probably the best, is the plant cannabis, which offers us not just one, but so many different compounds that can interact with this endocannabinoid system, as well as other targets in the body. And so we'll start with THC, which is a mimicker of our own cannabinoids, anandamide and 2-AG, by acting as a partial agonist at CB1 and CB2 receptors. Just some of the pharmacologic effects it can produce: analgesic, antipruritic or anti-itch, antispasmodic, antioxidant, bronchodilatory, neuroprotective, anti-inflammatory, anti-nausea, psychoactive. There's many other targets in the body. This is just a small list because anything that CB1 or CB2 can do, THC can do with it. There's other targets for THC in the body. It's not just working on CB1 or CB2. And, some of these targets are better or less understood in how clinically significant they may be. But you can see some other G protein and nuclear targets and some of the TRP receptors, as well as some inhibition at some of the other membrane receptors. Now, moving on to CBD, close sister to THC, very similar molecule. If you look at their molecular structure, quite different activity, actually. It's also strange because there's a lot of overlap in what we see as the outcomes with CBD. They both can help with pain. They both can help with anxiety and mood and all sorts of overlap, but the mechanism is actually quite different in a lot of ways. CBD has a very low affinity for the CB1 and CB2 receptors. This is not thought to be its main mechanism of action. Unlike THC, which is a partial agonist, CBD is an allosteric antagonist of CB1 and CB2. What that means is that when something else is stimulating CB1, CB1 and CB2, like anandamide or like THC. CBD doesn't block that stimulation. It actually it fits with a different site on the receptor, and it's like it turns down the volume a little bit. So THC or anandamide still gets to do what it's meant to do there, but it does so a little less loudly. It's also active in a lot of other areas in the cells, probably more than 40 or more than 60 that we know of. Some of the most clinically significant I've listed on this slide. So it activates the 5-HT1 serotonin receptor. This is thought to be the mechanism of its mood and anti-nausea effects. It activates the TRPV1 through 4 receptors, the PPAR gamma nuclear receptor, GPR18, the glycine receptor, which has a major role in the nervous system, antagonist of many other receptors, and then an inhibitor of the uptake of a variety of neurotransmitters, including anandamide. And so that's thought to be one of its mechanisms of action is through the inhibition of anandamide reuptake. And in the bullet point below that, you'll see also the inhibition of the Pha enzyme, which breaks down anandamide. These are 2 ways in which CBD can increase the levels of anandamide in the synapse or the circulating anandamide. And that could have indirectly a THC-like effect. CBD is also active in the mitochondria and much more. Here's a little graphical summary of some of the main effects of CBD that have been studied: the antispasticity, antidepressive, anti-anxiety, antipsychotic, anti-seizure, and anti-inflammatory properties. Now, I just had to say anti 6 times, which is one of the big problems with medicine and kind of research and how we think about these things, because, instead of saying antispasticity, we could say muscle relaxant or mood enhancing, or anxiety and fear reducing, or that would be anti. What can we say about that? Like safety and trust promoting, clear thinking, and so forth, balanced immune system. Moving on, THC and CBD seem to work really well together. CBD has been shown to decrease the undesirable side effects of THC, like intoxication, sedation, rapid heart rate. CBD can also enhance some of the benefits of THC, the analgesic, antiemetic, and also the anti-cancer properties of THC. It's also been shown to reduce the munchies that THC produces, which might be appreciated by some people or maybe not so much by others. There have been some studies, though, that have shown that even very generous doses of CBD do not necessarily decrease some of the psychoactive effects of THC. This study showed that it did not reduce at 0, 200, 400, or 800 milligrams of CBD taken orally before smoking really low-quality NIDA cannabis. In 31 people on average, there was no big effect on the reinforcing physiological or positive subjective effects reported by those subjects from the low-quality NIDA smoked cannabis. I think this tells the story a little bit more, though. Clinically, here we have a comparison of the toxic psychosis threshold. So we're not talking about long-term psychosis, but when you give enough THC to someone, they'll transiently experience psychotic symptoms. And if you look at the studies on dronabinol, or brand name Marinol, this is just a synthetic THC single molecule drug. The threshold on average is somewhere around 18 milligrams of THC. Someone who's not used to THC, give them around 18 milligrams. That's when you can start seeing some of these psychotic effects. Compared to Sativex, or nabiximols is the brand name, this is a 1-to-1 roughly oral spray of THC and CBD. The threshold seen in those studies for toxic psychosis was up around 48 milligrams of THC. So more than double the THC was tolerated for this very severe side effect when given with roughly equal portion of CBD. Now, there are many other active constituents in cannabis. I've chosen a few more to talk about. It would, again, take us all day to cover all the interesting ones. There are terpenoids, there's flavonoids. The terpenes or terpenoids give cannabis its aroma. They have therapeutic effects. They've been studied, as many of you know, from the essential oils of other plants. Then there's the acidic cannabinoids, THCA, CBDA. These were long thought to be inactive precursors. Now we know they're very active, just in different ways. And then also, many of these compounds are antioxidant and antimicrobial. You know, it's one thing to think about what are their targets in the human body, but also to think about what the heck are they doing on the plant. Well, many of them are protecting the plant from infections and from pests and from ultraviolet light and from oxidation. So we'll go through just a handful of these, starting with CBG, which is recently available from hemp extracts. I think in the last couple of years we've seen CBG products become more popular and more clinically recognized and utilized. CBG readily crosses the blood-brain barrier. On CB1 and CB2, it's a little bit more like THC, but not as strong. It's a lower affinity by a factor of 5 to 27-fold, so similar activity but not strong. Even high doses of it don't produce euphoria or strong psychoactive effects. It's also comparable to CBD at some of the ion channels. So it kind of looks like THC but weaker on some of the targets, looks like CBD on some of the other targets. One of its most interesting targets, I think, is this alpha-2 adrenoceptor, which is in the brain. It's part of the sympathetic nervous system, and stimulating this receptor decreases sympathetic nervous system activity. Similar mechanism of action as the drugs clonidine, which has many targets, but that's one of the things clonidine does, as well as guanfacine, which is used. So clonidine is a blood pressure medication, but it's probably the most off-label used drug around. It's used for treating withdrawal from opioids and benzodiazepines and alcohol. It's used for agitation and aggression and autism and in many other areas of medicine. Guanfacine is used in attention deficit hyperactivity disorder, and so as well as blood pressure, PTSD, dementia, and so forth. Therapeutic potential of CBG reviewed in a book chapter, which I found very helpful. And again, you can see some of these just wide-ranging conditions, including antibacterial. Many of the cannabinoids have been shown to be antibacterial. CBG seems to be the strongest. Let's move on to THCV, which is starting to become available. THCV is a CB1 antagonist at low doses and a CB1 agonist at higher doses. So it kind of flips its activity depending on how much is consumed. It's been shown to produce weight loss, to decrease body fat and serum leptin concentrations with increased energy expenditure in obese mice. It has some anticonvulsant properties. It's been shown to decrease swelling and sensitivity to pain and to lack the adverse effect profile of the inverse agonists like rimonabant. So rimonabant, a CB1 blocker, was actually approved in Europe and about to be approved in the United States as an anti-obesity, anti-munchie drug until there were too many side effects, including suicides. THCV is a gentle blocker of the CB1 receptor. It doesn't block it strongly like rimonabant, and it doesn't seem to have any of those side effects. I mean, go figure that the plant would also produce something that has that benefit without those side effects. THCA I found extremely interesting in my practice. It is pharmacologically distinct from THC, even though the only difference is that one carboxylic acid group. It has little to no activity at the CB1 or CB2 receptor and does not seem to have any psychoactivity, but it has been shown to modulate the immune system, having anti-inflammatory effects, strong neuroprotective effects, even though barely any of it gets into the brain under normal circumstances. That could be increased during injury or disease. It's been shown to suppress tumor necrosis factor alpha, independent of CB1 or CB2. That's a major player in rheumatologic and inflammatory conditions, to scavenge free radicals, to inhibit breast and prostate cancer cell lines. So many functions of THCA. I'll go over a few more. Anti-inflammatory, anti-fibrotic, meaning anti-scarring, in animal models of chemically and obesity-induced injury, including hepatic injury. It's been shown to reduce adiposity and prevent metabolic diseases via PPAR gamma. It's been shown in one study to kind of do the opposite of what CBD does, but to actually turn up the volume a little bit on the CB1 receptor. This is a positive allosteric modulator effect, which is very interesting. We've been seeing people clinically for years now claiming that products that are not fully decarboxylated, that have some THCA in there along with the THC, are more potent than the ones that have higher levels of THC. And I've wondered what is that? And I've thought, well, maybe the THCA is inhibiting the metabolism or breakdown of THC, and that could be a part of it, but it might also be because it's turning up the volume on the CB1 receptor as a positive allosteric modulator. And then in vitro and ex vivo models of inflammatory bowel disease, THCA has worked well in those models as well, without the biphasic pro-inflammatory effects of CBD. CBD is often biphasic, meaning one dose does one thing, another dose does the opposite, but that opposite often happens at super high doses that we don't really see in humans. Anyways, in these models of inflammation in the gut, the CBD at too high of a dose can actually have a pro-inflammatory effect. Doesn't seem like that happens with THCA. Now, anytime we think about THCA, there's always going to be a little bit of THC in there. It's 99% pure or 99% pure. You can never have zero THC in the presence of THCA. There'll always be a little. And maybe we think, well, that's so little, that probably doesn't mean anything. But I wonder, because I've seen some very interesting things happen with tiny, tiny doses of THC, almost like homeopathic doses of THC that you wouldn't believe, wouldn't expect. And so I wonder how often are these very low doses of THC a confounder in what we think we know about THCA. At this dose, 0.002 milligrams per kilogram of THC, I'm not sure what the equivalent would be for humans, maybe a ninth of that. That might be like, rubbing a little THC on your lip or something like that, like a tiny little piece of the plant. I mean, it's a really small dose. That dose has been shown to produce cardioprotection in mice that were given a heart attack, protect the liver from ischemia reperfusion injury in mice, and after a single dose, produce long-lasting activation of protective signaling molecules in the brain. So maybe a little THC and a very little THC and a little THCA together is a great combo, and there's no way to separate them. But we just don't know exactly what's happening there. CBDA is interesting. I've been very impressed with it clinically. Whereas THC and THCA are very different, CBD and CBDA are very similar. It shares the ability of CBD to activate the 5-HT1A serotonin receptor, but much stronger. It does not seem to act upon CB1 or CB2 receptors, so for some people, they don't want CBD to diminish the effects of the THC. They want the THC to really shine through. CBDA would allow that to happen. You could gain the benefits of CBD without inhibiting any of the effects of THC. It acts upon many of the same receptors, but it's been shown to be stronger at inhibiting the COX-2 enzyme, which produces inflammatory molecules, than CBD, THC, or THCA. It's also very well absorbed. The print is small. This is my last slide, so we'll wrap it up in just a minute here. But I wanted to point out how bioavailable this stuff is. First of all, CBD, in a model of nausea, CBD demonstrated anti-nausea properties in rodents at 5 milligrams per kilogram. CBDA was effective at 0.0005 milligrams per kilogram. And the dose-response curve was not biphasic, which it was for CBD. Go from 5 to 20 milligrams per kilogram of CBD, and you go from anti-nausea to pro-nausea effects. That was not seen to happen in the doses studied of CBDA. So, I mean, how many? 1, 2, that's like a thousandth, like 1,000 times more potent than CBD in that model of nausea. Again, you can see here 100 times more potent than CBD in a model of increased sensitivity to pain, and 10 times more potent than CBD in a model of seizures based on the Dravet syndrome. So, and barely any CBDA gets into the brain, but it's still 10 times as potent as CBD in reducing those seizures. I didn't show it here, but the bioavailability in humans has been studied of CBDA versus CBD, and CBDA is at least 10 times, but up to 30 times more bioavailable when taken by mouth. So you're getting a lot more of it into your blood, and then it's having a much stronger effect. So that is the summary of the endocannabinoid system and cannabinoid pharmacology. I know it was a lot. That was a whole hour. Be sure to get up and stretch. And thank you very much.